A carbohydrate is a biomolecule consisting of carbon C, hydrogen H, and oxygen O atoms, usually with a hydrogen-oxygen atom ratio of 2 to 1, as in water. The empirical formula is CH2O(N). They are defined as polyhydroxyaldehydes or ketones, or those compounds which produce those products on hydrolysis is called carbohydrates. Act as a precursor of many organic compounds. Their other forms are glycoproteins and glycolipids on cell membrane, helps in cell growth and fertilization.
They are the hydrates of carbon. Storage form of energy is glycogen is used under the starvation condition. Saccharide, a group that includes sugars, starch and cellulose. Carbohydrates are the sugars, starches and fibers found in fruits, grains, vegetables and milk products. Carbohydrates, one of the basic food groups important to a healthy classification. Carbohydrates are commonly referred as saccharides. Greek, saccharin means sugar. Most carbohydrates are polymers. Polymers are large, complex molecules composed of long chains of monomers. Monomers are small, basic molecular units. Carbohydrates are classified into three major groups depending upon whether these undergo hydrolysis, and if so on, then the number of products formed by them, and based on number of structural units. They are monosaccharides, oligosaccharides, monosaccharides. Monosaccharides are sugar molecules containing short chain of carbon atoms, one aldehydic or ketonic group and hydroxyl groups attached to remaining carbon atoms. If they contain aldehyde group, they are named as aldoses, and if they contain ketone group, they are named as ketoses. They are the simplest carbohydrates, cannot be hydrolyzed to simpler units. It ends with -ose. Example, glucose, fructose. 4 C atoms, pentoses, 5 C atoms, hexoses, 6 C atoms, and so on. Examples are glyceraldehyde, erythrose, ribose and glucoheptose. Oligosaccharides, they produce 2-10 molecule of monosaccharides on hydrolysis. Empirical formula is CnH20n1. Oligosaccharides are formed by polymerization of monosaccharide molecules by elimination of water molecules. The linkage between monosaccharide units in oligosaccharide units is glycosidic linkage, depending on number of monosaccharide units. Oligosaccharides are saccharides etc. classified as D-, tri, tetra-. Examples are lactose, raffinose and stachyose. Polysaccharides produce more than 10 monosaccharide units on hydrolysis. They are high molecular weight substances composed of large number of monosaccharide units combined to form one large polymer molecule. They may be straight chain or branched chain polymers. Based on the type of monosaccharide, they are classified into homopolysaccharide. Produce same type on hydrolysis. Example is starch, chitin. Heteropolysaccharide, produce different type on hydrolysis. Example is heparin. Hyaluronic structure. The carbohydrates can be structurally represented in any of the three forms: open chain structure / Fischer projection. It is the long straight chain reform of carbohydrates. The L and D conformations represent the absolute configuration of the asymmetric carbon farthest away from the ketone or aldehyde group on the monosaccharide. On the Fischer projection, if the farthest hydroxyl, OH, group is on the right, then it is classified as D sugar. If the hydroxyl group is on the left, then it is Al sugar. Hayworth structure, it is the presence of the pyranose ring structure, hemiacetal structure. Here the first carbon of the glucose condenses with the OH group of the fifth carbon to form a ring structure. Fischer projection discovered by German chemist Emil Fischer in 1891. In a Fischer projection, the carbohydrate is shown in its open chain form, rather than a cyclical one. Carbon atoms in the main chain of the carbohydrate molecule are connected vertically, while hydrogen atoms and hydroxyl groups horizontally are bonded. Fischer projections have one main advantage: it is easy to visually identify the stereochemical properties of a carbohydrate and compare the difference between two carbohydrates quickly and easily. For example, it is simple to tell the difference between two enantiomers. Hayworth projection. A Hayworth projection differs from a Fischer projection in that it is used to represent the carbohydrate in its cyclical form. This is especially useful for sugars which have a ring structure. It was devised by the English chemist Sir Norman Hayworth, who expanded on the work of Fischer, characterizing many more carbohydrates. A Hayworth projection is a simple way to show cyclic sugars and their glycosidic linkages. If drawn from a Fisher projection with the carbonyl on top, the groups on the right side become the groups on the bottom of the ring and the groups on the left become the groups on the top. The carbon at the very bottom of the Fisher projection is placed on top of the ring by default. If it is not a part of the ring itself, an A linkage occurs when the hydroxyl of the hemiacetal is on the bottom of the right and a linkage occurs when the hydroxyl is on top. Metabolism. Metabolism is the total amount of the biochemical reactions involved in maintaining the living condition of the cells in an organism. All living organisms require energy for different essential processes and for producing new organic substances. The metabolic processes help in growth and reproduction and help in maintaining the structures of living organisms. The organisms respond to the surrounding environment due to metabolic activities. Chemical reactions occurring in the living organisms from digestion to transportation of substances from cell to cell require energy. There are two types of metabolic process: catabolism, anabolism. Catabolism. This process is mainly involved in breaking down larger organic molecules into smaller molecules. This metabolic process releases energy. Anabolism. This process is mainly involved in building up or synthesizing compounds from simpler substances required by the cells. Glycolysis is glycose sweet or sugar, lysis dissolution, a universal pathway in the living cells. Glycolysis is the central pathway for glucose catabolism, in which glucose, 6-carbon compound, is converted into pyruvate, 3-carbon compound, through a sequence of 10 steps, with the production of ATP. The complete pathway of glycolysis was elucidated in 1940 by Emda Meyerhoff-Parnasse and is frequently referred to as EMP pathway. Glycolysis takes place in all cells of the body. The enzymes of this pathway are present in the cytosomal fraction of the cell. Glycolysis occurs in the absence of oxygen, anaerobic, or in the presence of oxygen, aerobic, lactate is the end product under anaerobic condition. In the aerobic condition, pyruvate is formed, which is then oxidized to CO2 and H2O. Glycolysis is a major pathway for ATP synthesis in tissues lacking mitochondria, e.g. erythrocytes, cornea, lens, etc. Phases of glycolysis. During glycolysis, the 6-carbon glucose is broken down into 2 moles of 3-carbon pyruvate via 10 enzyme-catalyzed sequential reactions. These reactions are grouped under 2 phases. Phase 1 and 2. A. Phase 1 or preparatory phase. It consists of the first 5 steps. In these reactions, glucose is enzymatically phosphorylated by ATP, first at carbon 6 and later at carbon 1, to yield fructose 1,6 diphosphate, which is then split in half to yield 2 moles of the 3-carbon compound, glyceraldehyde 3-phosphate. The first phase of glycolysis, thus, results in cleavage of the hexose chain. This phase requires an investment of 2 ATP moles to activate, or prime, the glucose mole and prepare it for its cleavage into 2 3-carbon pieces. Besides glucose, other hexoses such as D-fructose, D-galactose and D-mannose may also convert into glyceraldehyde 3-phosphate. B. Phase 2 or payoff phase. The last 5 reactions of glycolysis constitute this phase. This phase represents the payoff of glycolysis, in which the energy liberated during conversion of 3 moles of glyceraldehyde 3-phosphate to 2 moles of pyruvate is converted by the coupled phosphorylation of 4 moles of ADP to ATP. Although 4 moles of ATP are formed in phase 2, the net overall yield is only 2 moles of ATP per mole of glucose oxidized, since 2 moles of ATP are invested in phase I. Thank you.